Welcome to Part 4 of Chapter 9, the Muscular System. This video focuses on the electrical component of muscle contraction. We will discuss ion channels and membrane potentials. We will also examine action potentials. Let's begin with ions and membrane potentials. Like neurons, muscle fibers are electrically excitable. Action potentials travel from the brain along the axons of neurons to our muscles, where they cause contraction. Let's learn how this happens. At rest, muscle cells are polarized. Polarized means that the inside of the cells are more negatively charged than the outside. This image shows an oscilloscope measuring both the inside and the outside of a resting skeletal muscle cell. It reads negative 85 millivolts. Negative 85 millivolts is the resting membrane potential of a muscle fiber. As you may remember, ions are positively or negatively charged atoms. Their concentrations and location determines the membrane potential of a muscle fiber. Ion channels are located in the plasma membrane, and they control where the ions are located and the cell's membrane potential. The three types of ion channels are leak, ligand-gated, and voltage-gated. Leak channels are always open, and ions flow through the channel based on the concentration gradient. The image on this slide is of a leak channel. Ligand-gated channels open when a specific chemical, like a neurotransmitter, binds to it. They are closed when the chemical is not attached. Voltage-gated channels open and close depending on the specific membrane potential. All three of these ion channels play an important role in creating action potentials and muscle contraction. Let's move on to learning about action potentials. Action potentials in muscle fibers occur when a skeletal muscle is stimulated by a motor neuron. In an action potential, the membrane potential changes so the cell becomes more positive inside than outside. As a reminder, during resting membrane potential, the inside of the cell was negative 85 millivolts, so it was more negative than the outside. Action potentials in muscle fibers are due to the ion channels opening. This allows ions to move across the plasma membrane and thereby changes the membrane potential of the cell. As a reminder of where ions are normally located, remember the salty banana. Bananas have a lot of potassium in them, and salt has a lot of sodium in it. So if you think of a banana covered in salt, you would think that inside of a cell there is more potassium than outside, and outside the cell there is more sodium than inside the cell. This situation creates concentration gradients for both sodium and potassium. Because of the concentration gradients, ions will flow from areas of high concentration to areas of low concentration when ion channels open in the plasma membrane. Action potentials have two phases, depolarization and repolarization. At rest, the cell is polarized, and its resting membrane potential is negative 85 millivolts. During depolarization, the inside of the cell becomes less negative. If the cell depolarizes enough to reach the threshold value, an action potential begins. Further depolarization occurs and the membrane potential reaches positive 20 millivolts. The second phase, repolarization, is the returning of the membrane potential back to the resting membrane potential. So in this phase, the membrane potential goes from a positive 20 millivolts to negative 85 millivolts. The image shows the changing membrane potential a muscle cell goes through. I recommend pausing this video now to take a good look at the graph before continuing. Lastly, remember that it is the opening and closing of ion channels that cause all of these changes in the membrane potentials. So what exactly is happening to the ion channels? Let's investigate what the different channels are doing that cause this action potential. The lower left image shows what the plasma membrane of the muscle fiber looks like. At rest, sodium and potassium voltage-gated channels are closed. Potassium leak channels are open, so potassium leaves the cell through them due to there being more potassium inside than outside the cell. The result is a membrane potential of negative 85 millivolts. As we go through each step, pause the video until you have digested the information and are ready to move on. It is not uncommon to have to go through these steps several times until you really understand it. When the muscle cell is stimulated, sodium channels open. Depending on the stimulus, it can be ligand-gated or voltage-gated channels which open, but it's always a sodium channel. Due to its concentration gradient, sodium enters the cell, making the inside more positively charged. This is depolarization. If an action potential is to be created, the membrane potential moves from negative 85 millivolts to the threshold value. Once the membrane potential reaches threshold, many voltage-gated sodium channels open rapidly. This causes sodium to diffuse into the cell, and this results in more depolarization. The membrane potential increases from the threshold to positive 20 millivolts. If the threshold was not reached during the initial depolarization, an action potential would not occur. Once the membrane potential reaches positive 20 millivolts, voltage-gated sodium channels close and voltage-gated potassium channels open. Potassium leaves the cell, making the inside more negative. This is called repolarization. At negative 85 millivolts, all voltage-gated channels close. Because the voltage-gated potassium channel close slowly, the membrane potential briefly dips below negative 85 millivolts. This is called hyperpolarization. The sodium-potassium pump reestablishes the resting ion balance. Action potentials in one area of the muscle fiber propagate along the entire muscle fiber. The green area in the image is where the action potential is occurring. Note that the green area inside of the cell is more positive than the outside. This is reversed from what happens at rest. Membrane depolarization in one area triggers the opening of voltage-gated sodium channels in the adjacent plasma membrane. The adjacent area becomes depolarized and an action potential occurs there. The action potential continues or propagates along the entire plasma membrane. This video covered the electrical components of muscle contraction, including ion channels, membrane potentials, and action potentials.